PCR is an incredibly important technique in biology, and nowhere more so than synthetic biology. It's used for a variety of applications. It can be used to screen transformants. It can be used to amplify out sequences for cloning. There's a number of things you can do with it. It's very important. But how does it actually work? And in order to understand that, we're going to need to delve into the theory. So let's start with the aim of PCR, or polymerase chain reaction. So initially in PCR, you have template DNA. This looks something like that. My full line represents the amplicon that I want, the DNA sequence that I want to amplify for whatever reason it is. And my dashed lines are going to represent junk DNA, DNA on either sides of my amplicon in the template DNA that I just don't want. And over the course of the 30 to 35 cycles that the PCR machine will do, I want to take that, the very little amount of template DNA that there is, less than 1 nanogram usually, and I want to turn it in to a bunch of new strands, which I'll draw in red. And each of these strands should look like just the double-stranded amplicon. So in the original template DNA, it is double-stranded. It does have to be double-stranded for this technique. I've drawn 4 here. In practice, we'd be looking at more like 4 times 2 to the 32. So it's a lot of DNA that you want to generate. And that's very powerful if you can get that to work. But let's look at the theory of that. And the way we're going to do that is by going through a complete cycle. In fact, complete 3 cycles, I think, is what we'll do. So I'm going to draw template DNA in black. I'm going to draw new DNA in red. I'm going to draw primers in blue. So initially, we'll say we start with 1 template DNA strand. In practice, it's more than 1. It's quite a lot, but its relative amount we'll say is 1. So double-stranded DNA, template DNA. The very first step in PCR is to lyse the cells with heat. Having done that, the heat stays for long enough to make the double-stranded DNA de-anneal. So this will now be 2 single strands of DNA. The temperature then cools such that the primers can anneal. In theory, it cools enough that the template DNA can re-anneal with itself, but we've got many million times more primers in here than we do template DNA. So it's much more likely to happen with a primer. So I was going to draw primers in blue. Where do the primers actually sit? Well, the forward primer sits on the reverse strand, right there. The reverse primer sits on the forward strand, the opposite strand, going the other way. And when we perform an extension reaction about 72 degrees, the result we can determine completely logically. So, what does it look like? I'm going to go through each strand one by one. So, the first strand I say is my template strand. Nothing happens to this template strand. It stays the same. So I still have it. The next strand I say is I have a primer, and that primer is going to run from where it is. So it's going to amplify from the start of where it is over here. I said I can draw some red, so I'll draw it in red. So it runs from the start all the way to the end. And that would be brilliant, except it doesn't actually do that. It keeps going. There's no reason for it to stop. It doesn't know that that's the end of your amplicon, so it just keeps going. It won't go forever because the extension time is only 1 to 10 minutes usually, so it will only amplify really a maximum of 10 kilobases, but it will keep going way past what you actually want your insert to be. So how does that work? Well, we'll come to it. But for now, let's keep looking at these strands. So the third strand I see is from this primer here. So that one will start at the left-hand side of the amplicon, 5' side. It will run to there, and as before, it doesn't know to stop. So it just keeps going. Okay, last strand is the other template strand. So after one cycle of PCR reaction, we see we have 0 copies of what we want, which is certainly a result. But let's see what happens if we keep going. So I'm going to add more primers. So there will be a reverse primer will sit on this strand, as before. A forward primer will sit on this strand. These strands do actually have different polarities, and I haven't noted them down here. But if you follow through how they're created, you'll see the polarities as you go. This strand will be a reverse primer at the top, and this one will be a forward primer at the bottom. You can also kind of see that these two were made together, so they would re-anneal. Well, they would be annealed during the extension, and these two were made together, so they would be annealed during the extension. So the polarity is conserved. Okay, let's look at what happens when we add these primers. So again, we're going to go through strand by strand. The first strand I see is template strand. And what you may notice is we may not be making any new template strands, but they're sticking around. So we're not getting rid of the template strand. It's just sticking around doing nothing. Then the next strand I see is, as before, this reverse primer on top. So that's going to make the same as I saw last time. It's going to be amplicon, and it doesn't know to stop going. So it's going to keep going. Okay. The third strand I see is this second primer here on top of the red strand. And this is where it starts to get interesting, because this one only starts at the start of the amplicon, and the end of the amplicon is right there. So it doesn't have any junk DNA to keep transcribing, so it will only transcribe the amplicon. So now I've got a strand that's the amplicon. Now my next strand I see is this strand that was created in the last one and is now serving the template strand. Okay, template strand doesn't disappear, so it's still there. Next strand I see is another template strand, so that stays the same. I will just mark in green which of these are pairs. So those are pair, that's a pair, and by pair I mean they were extended together. So the next strand I see is this blue primer here. Again, this starts at the start of the insert, start of the amplicon rather, and it extends all as far as it can go, but it ends as soon as the DNA ends, and the DNA ends at the end of the amplicon. So we have another correct size amplicon. I'll just extend those so they look similar. So again, those are paired. And if I do the last one, last two, sorry, then I see, as in the first cycle, there's a primer here, which will generate a long, junky one. And then I have my very first template strand that was a reverse strand, so that will stay the same. Cool. So after 3 PCR cycles, we still have 2 template strands. We have 4 half correct amplicons, and we only have 2 of the ones we actually want. So this isn't very promising so far. Doesn't look like we're making much progress. Let's see what happens when we do one more cycle. If I add primers onto all of these again, you can see there's a pattern to the way I'm doing this now, so I can see how they're paired. I can see what the polarities therefore are. Then I'm just gonna make a new section to work with. Okay, this will be after our third PCR cycle. I'm going to draw all the strands I see, starting at the top. One template strand. Next one is a long junky strand. Okay, I'm in the way. So I'll move over here. So next one I see is correct strand was a template for this round. Nope, I'm missing one. I'm missing those two there. So I see, yes, I see a correct strand. And then I see a template long strand, half long strand. And then I see a correct strand, which was a template for this round. And I see a primer, which is going to amplify the correct amount. Then I see a primer, which is going to amplify the correct amount. And I see a long junky template strand. Then I see, oh, I'm in the way. I see a long junky template strand, which I'll start drawing over here now. So this is this red one here. And I see a primer that's going to amplify the correct region. And then I see a primer, which is going to amplify the correct region. And then I see a correct region already as a template. And then I see a long junky template strand. And I see a primer, which is going to amplify the correct region. And I see a primer, which is going to generate a long junky template strand. And I see one other template strands. So let's count up how many correct amplicons we have now and compare them to how many we don't. So of template strands, 1 and 2, I still only see 2 black ones. So they don't grow. That remains the same the whole time. We do still see growth of the half correct amplicons. So now I have 1, 2, 3, 4, 5, 6 of these. So we can see a pattern here. 0, 2. There's 4 in that one. And then there's 6 in this one. You can see, it's grown by 2 each time. And that is actually correct. It does grow by 2 each time because each time template strands can be used to generate 1 each. But now let's look at the amplicons. So I went from 0 to 0 to 2 to 1, 2, 3, 4, 5, 6, 7, 8. 8's there. So I've gone 0, 0, 2, 8. And if I keep doing this, it does begin to grow exponentially. And it is a massive, massive increase. It does double every time when you get to this sort of stage. The reason it doesn't look like it's doubling is because we've got these minor contributors still going. But when it gets to the case that these correct amplicons are more prominent, it does begin to effectively double each time. So PCR, very powerful technique, not without its limitations, but incredibly important, and it's well worth understanding how it works.